I'm here today to tell you a little bit about how the GMO testing actually works at a very high level from the biology, and then more specifically about the testing technologies and how they're used to enable the non-GMO markets. So I'll jump in, and please type any questions in the chat. So there are many challenges for detecting GMO in corn. Some of these come from the market. So there's different end customers that have different requirements. So you really got to know who you're selling to and what their requirements are. From a grower's perspective, it's, what elevator or processor am I selling to? And really understanding what type of test they're going to hold you accountable to. And if they have a specific threshold that they're looking for is very important for the grower. So for example, the non-GMO project, they have a 5% cutoff in feed. However, if you're selling to a feed mill directly, they may have a 3% cutoff because they want to create some room for error when they actually go to aggregate the grain and transform that into a finished feed. So it's very important for you to know your buyer and know what they're testing with and know their thresholds so that you can be aligned with their testing protocol, so there are no surprises when you go to make that delivery. The next challenge is, when we think about GMO testing, it's to support the entire supply chain that enables that value-added product that has that differentiated label and that claim that's backed up by testing. So the type of tests that are available, the DNA tests or the protein tests, they have different characteristics that lend themselves for use at different areas of the supply chain. So some of these areas quickly could be testing of the corn seed itself for purity, testing at a probe stand to judge purity before putting it into a bin. So you have your options depending on what type of test you want to use. And those options, and usually the test is determined by the need for speed. And we'll go into a little bit more detail around what's the appropriate test by the use occasion. And another challenge with corn is that there are a lot of traits that are commercialized, multiple modes of herbicide tolerance, multiple modes of insect resistance, and multiple quality output traits, whether that's drought tolerance or expression of amylase from anagen corn. So there's a lot of different traits out there. The actual end markets, whether it's an export or the non-GMO project, they may have different requirements for what they actually test for. So being able to develop a test that is comprehensive and supports the particular label can be difficult when it comes to making it cost effective. And so this is very high level challenges, and we'll start with a few slides that take a high level approach of explaining what's actually happening with the GMO, and then build in the complexity, take a look at the testing technologies themselves, and ultimately what you should do as a grower to make sure that you receive the premium that you worked hard to get. So what is a GMO, genetically modified organism? The goal of creating GMOs is to overall increase your productivity, increase your yield per acre. How this is achieved is that by looking at the traits of other organisms, so bacteria or fungi, they may find some genes, which is DNA, that express certain proteins, which is shown here in green, that have interesting properties that a plant could take advantage of if the plant were to express that protein. So for example, Roundup Ready corn, that gene was taken from a different organism. It expresses a specific protein, and what that protein does is it deactivates glyphosate once it's sprayed on the plant. So therefore, it enables tolerance of glyphosate. So DNA, there are DNA tests and there are protein tests. The DNA test is called PCR. It just stands for polymerase chain reaction. It has a long time to result, and it requires some sophisticated instrumentation to be able to run that analysis. The proteins can be detected by strip tests. It's very similar technology as the pregnancy test, where you run a fluid up a porous membrane, and there's some binding that occurs, and you see a line develop. And with protein tests, the intensity of the line correlates with the amount of protein in the sample, which therefore correlates with the amount of contamination on a percent weight basis. So it's important to note that the two type of tests that's used by the supply chain are looking for different actual substances. The DNA test is looking for that foreign piece of DNA, and it's amplifying and detecting that specifically. Whereas the protein tests look for the protein that's expressed by the DNA, it's coded for by the DNA, and it quantifies the amount of protein present. And depending on where you are in the supply chain, you may favor the protein test because it's fast, or you might not be able to use the protein test because you've started to process your corn, and now the proteins have changed shape considerably, and the protein test will no longer work. So if you think of testing anything that's been heat treated or closer towards that end product, say it's a tortilla chip, the protein test will no longer work because that corn has been significantly processed to the point where these proteins no longer look the same. A little bit of nomenclature. There are traits that are monotrait lines, meaning there's only one GMO in the trait or in the seed. An example of that would be Roundup Ready 2 corn. It usually has one trait associated with it, such as herbicide tolerance. And then there are stack traits, which just means that there's more than one GMO trait being present. So an example of that would be BT Double Pro. Trait stacks can be only herbicide tolerance, so where you have multiple modes of action for various herbicides. So that could be Roundup and Liberty stacked because you don't have insect pressure. Or it could be with multiple modes of insect resistance. And over the years, the adoption of stack traits has significantly increased. And the need for that is based on the evolution of weeds and the actual pests of the cornfield. They're developing tolerance to only one of the specific herbicides or multiple forms of the herbicides. So one strategy to try and keep those fields clean and the harvest or the yields high is to stack them up, and they tend to work better and eliminate some of those resistances. So as we progress from monotrait to stack traits, the stacked lines tend to pose a little bit more difficulty with making sure that the GMO or the protein tests for GMO align with the DNA tests for GMO. And I have some additional slides to show why that is the case about a few minutes from now. So the first GMO testing technology we'll take a closer look at is DNA detection. So once again, the actual method is PCR, polymerase chain reaction. And for the sake of the grower, if you ever want to get this type of analysis done, you'll likely be sending it to a laboratory that can perform this analysis for you. Envirologics has a lab service, and we offer DNA detection. It generally takes 2 or 3 days for you to receive results. The GMO genes are quantified by comparison to known standards. DNA detection is considered the gold standard technology by some of the common non-GMO labels, such as the Non-GMO Project, for example. And it's used at a point in the supply chain where you're closest to that finished material. And it's the gold standard because only the DNA detection will work once you start processing the corn and get closer to that finished good. And the consumer wants to be sure what they're actually consuming is non-GMO. So that's why DNA is considered the gold standard for that particular non-GMO program. In general, the DNA tests are much more expensive than the protein tests, about 10 times. So the protein detection, this is our product over here. We call it a comb. So a comb is just it illustrates the actual configuration. So it's multiple strips where each strip detects a specific GMO protein that is being cultivated in corn. And we call it a comb because if you think of each strip being a prong on a comb that you brush your hair with, it's just a shorthand way that we can describe the product as one unit. And when you're actually doing the testing, it makes it a lot easier to handle one unit instead of having to thumb through 10 individual strips. So these detect the proteins. It's a 5-minute runtime, similar to a pregnancy test. Since they're so fast, they're great for screening grain at delivery points or critical control points that help you segregate the grain based on the contamination level. So how these work is that this barcode contains a standard curve where we have a certified reference material in-house for each individual trait. And we look at the line intensity. The darker the line, the more of the GMO protein present in the sample. And the reader, it's called the quick scan that we offer. It basically measures the line intensity, the reflective units off that line, compares it to the standard curve that each manufactured lot gets built around, and then it assigns a % contamination level based on that line intensity. We have some example reports that we can talk through over the next couple slides to give you a closer look about how that report is actually generated and how to interpret the report. So if you're looking to send out for DNA analysis, the process would look a little bit like this. First, you'd call in your order. They'll help you with the right product order, the right catalog number, etc., where to ship the product, how much of the sample to ship. Then it would arrive at a laboratory. There would be some processing that would be done. Particularly, it's extraction of the DNA and purification of that DNA. And then there's the actual PCR test that occurs where that DNA is amplified, detected, and quantitated against the standard curve. A standard panel, this is one that's used by the Non-GMO Project right now for GMO corn detection. It includes 4 individual tests. So similar to how the protein tests has 10 strips, 1 for each specific trait, the standard GMO corn panel has 4 individual tests, and these tests are summed up to create a GMO sum. And then that GMO sum is what you'll be compared to by either the Non-GMO Project itself or whatever the non-GMO program or export customer requires to be under for a GMO sum. So this is just a standard panel. It can change depending on your customer, depending on the buyer and the non-GMO program itself. So the names are going to look different too, because they're detecting events and not the protein. So for example, or they're detecting genetic sequences rather than the protein. So MIR162 expresses a specific protein. I can't remember off the top of my head if it's VIP3A or if it's modified CRY3A, but that encodes a specific protein. Whereas some of the other DNA tests like CMV35S, that is a piece of the DNA strand that is commonly used across many of the GMO traits out there. So it can capture a lot of the contamination. There's a slide that compares the 10 traits on the comb to this PCR panel coming up, and you'll be able to see how it can capture various traits by using one genetic sequence. So what does the protein test look like? After collecting a representative sample, which to do it right takes some time, multiple locations in the truck need to be probed. You need to get a size which is representative, grind it, take a subset of that. Then you extract with water with a 30-second shake, transfer the extract into the cup, add the comb, wait 5 minutes for the test to develop. And then you put the test into the reader. So for Envirologics, that's the quick scan reader. You can also run mycotoxins. The reader also tests for mycotoxins. So if your buyer has a concern about mycos, they can run that and get the data alongside the GMO in one report. This provides individual line quantitation or individual trait quantitation, and then it takes a GMO sum of that as well. And that GMO sum is generally what you're compared to on the decision about whether they're going to accept the grain as non-GMO or have to accept it as commodity. So now we'll take a little bit closer look at what these reports look like and how they compare. So first off, these two reports do not represent the same sample. They're just, they happen to be screenshots that I had access to. And I just wanted to walk through these a little bit to show you what type of data would be generated. So I already walked through the DNA test a little bit, where you have the standard panel for corn has 4 individual tests that are quantified and then summed up into a value. And then the GMO corn comb has 10 tests on it that are individually quantitated and then summed up for a GMO corn sum value. So generally, depending on where you are in the supply chain, these GMO sums are going to be the ones that are compared to the acceptance level. And then the decision is made at that specific point, typically either with the protein test because there's a need for speed, or with the DNA test because it's a complex sample, it's been processed. And the only way to really find out if there's GMO in there is to run a DNA test. So we often get the question, do DNA and protein tests provide the same result for the same sample? And in some cases, yes, in some cases, no. It depends on how that sample is contaminated. But generally speaking, protein tests are a conservative screening tool. And they will provide results that are equal to or greater than the DNA test. And this ensures that bins are kept below threshold values and gives buyers confidence to aggregate grain and then process large batches of grain in non-production GMO and production non-GMO runs. So if you look at a data set, it doesn't match up 100% correlation. So for example, protein tests will give you a value that's less than the DNA result 20% of the time. Will give you a result that's in line with the DNA test 40% of the time. And we may slightly overcall sometimes, maybe 40% of the time. So we are actively trying to figure out how we can bring the protein test closer to the DNA test. And we just launched our lab service, and we're thinking that we'll be able to use, if our customers allow us to use their samples for R&D purposes, we'll be able to use that data that we're generating in our lab service to compare it to our protein tests, our combs, and come up with mathematical ways to normalize the two. So that's something that we'll be working on the next coming years, trying to to bring any discrepancy to a closer agreement. But fundamentally, why do they give different results? To answer that is a bit complicated, and it involves some advanced concepts from biology. But at a high level, the reason is due to all the stacking that occurs in GMO corn, and then how those stacks can contaminate non-GMO corn. So if you look down at the panel here, the DNA standard panel, this CAM V35S is a common genetic element that is in the genes for CRY1, CP4, CRY3, CRY1F, PAT, CRY34, and CRY2. So in some cases, there's contamination, say that it's a stack of herbicide tolerance traits. So you have your Roundup and your Liberty, which is the PAT. In this case, we would call that correctly. It'd be 1-to-1. But in other cases, there might be only 2 35S's present in the stack, but we're detecting 3 proteins. So we'll have a tendency to slightly overcall it. The problem is that we can't say for sure where this 35S is coming from. So that's why we tend to give a conservative answer. So that's a bit of a complicated explanation. I'm sure there'll be some questions on that. So I'm happy to come back to that after the presentation is wrapped up.
Okay.
So the right GMO test type depends on your need and the use occasion. So if you're just looking for a result, if you just want to send out to a lab, you actually have both options. You could do the DNA test or the protein test. So if I was a grower of grain, I would want to send out to a lab and get my grain screened by the protein test, knowing that the protein tests, so that's the combs or the strips, those are the ones that my buyer as a grower are going to use to evaluate the purity of my corn. I would ask for the same type of protein test if I'm buying corn seed to be done, because I need to know that whatever I plant is at the right purity level as determined by the protein test, so that there's no surprises when I go to sell it, assuming I don't get any contamination during the growing process or transportation process. So if you're doing a DNA, why you'd want to send out for a DNA sample is if you're primarily a processor, which comes down to some of these milling products in the finished food or finished feed itself. The protein test simply won't work there because the processing changes the protein shape too much and the results can no longer be trusted. Some of the other reasons why you would go with a protein test, it's because it's less expensive, about 10 times less. And the actual equipment, so the quick scan reader that reads the GMO and the myco strips that Envirologics makes, those are measured in the low thousands of dollars, where a PCR machine, a thermocycler, costs tens of thousands of dollars. So that plays a role in deciding what system to invest in. And then also the speed. If you're trying to segregate grain based on the point of grain delivery, DNA testing, it doesn't fit the use occasion or the need. So to sum that up, it's time to result, the sample itself, and the cost of the test that determine how I'm going to evaluate whether the sample contains GMO or not. But ultimately, in addition to these factors, there's also what you need to do to conform with any non-GMO program. So the non-GMO project requires PCR testing for these, for say, for example, corn gluten meal or any type of processed material to get that butterfly label. So there are some standards that, and also export customers that will say, I need the final screen to be PCR or a DNA test. So since this webinar is to support growers and make sure that they have good guidance on how to grow non-GMO and to earn that premium that they work so hard to get, We put together a quick slide to discuss that scenario. So the first thing that I think a grower should do is talk to their buyers about what are the thresholds that I need to meet and how are you going to test for that threshold. So once you understand that, then you can go to your corn seed dealer and you can say, these are the requirements I need to hit. Knowing that when I grow, I am subject to a little bit of risk of additional GMO contamination. I need some wiggle room in my purity level. So for example, if you're being held to a 0.9% GMO sum, then I don't think it would be a good idea for you to buy seed that is 99.1% pure, because that gives you no room for error during your growing process. You would want to go with something like 99.9% pure. However, if you're selling to a feed market that's non-GMO, and the threshold you're being compared to is 3% or 5%, then a 99.1% purity level on your corn seed should be okay. In addition, you should ask your seed dealer how they're assessing purity, knowing that ultimately you're going to be held to the protein test. So using more specifically the Envirologics corn cone, if you can figure out exactly what that test is, you could do some pre-screening of the corn seed itself to make sure you're at that right purity level before you invest all your time in planning it and growing it up properly. You'll want to follow the good guidance from Tony and Craig around best practices for keeping your cornfields clean. When you go to harvest it, store it, and transport it, you'll want to pay close attention not to have any commingling with GMO grain, and also GMO soy. So the protein tests, the strips and the combs, those detect traits or the proteins that are from GMOs, and some of those GMOs are also present in soy. So DNA tests can be specific enough to say the DNA I'm detecting is from corn and it's a corn GMO trait. The protein tests are not that specific. They just say, I see a GMO and here's the level that I see. It won't tell you whether that GMO comes from corn or from soy. So you got to be careful about having some commingling with soy as well as you store and transport your grain. And if desired, you can do some advanced testing before you invest in the freight to move your grain to the buyer. Finally, Nathan had asked for a few sources of seed for non-GMO corn seed, and we know that these companies here do a good job with their testing in general. So Blue River was what Tony had talked about before. They offer pure maize, but they also have other different types of non-GMO corn seed available. And Albert Lee they offer a couple different purity levels. They offer 99.9% pure and 99.1% pure. So once again, if you know what your thresholds are, you can make a better decision about your corn seed in terms of how much should I pay and what are the varieties available to me.